In the realm of science, where the boundary between the known and the unknown is constantly being redrawn, a phenomenon as eerie as it is fascinating has captured the attention of researchers worldwide. This phenomenon, known as zombie genes, refers to the peculiar behavior of certain genes that spring to life after the death of an organism. Far from the realms of science fiction and horror, the study of these genes opens new horizons in understanding life, death, and the potential for medical advancements. This article delves into the enigmatic world of zombie genes, exploring their discovery, implications, and the groundbreaking possibilities they present for science and medicine. The discovery of zombie genes has transformed our understanding of life after death, shifting the paradigm of biological cessation into a realm filled with unanswered questions and boundless curiosity. Initially, the concept of genes that activate post-mortem seemed more aligned with the plots of science fiction novels rather than the annals of scientific journals. However, as researchers delve deeper into the molecular activities that persist after an organism's demise, they uncovered a startling reality. Death is not the immediate end to biological activity that we once thought it to be. This realization came about through meticulous research and observation, where scientists noted that specific genes not only remained active but actually became more expressive after the organism had died. These genes, which had lain dormant or minimally active during the life of the organism, suddenly surged to life, earning them the moniker of zombie genes for their post-mortem awakening. The phenomenon was first observed in the context of studies aimed at understanding the degradation processes of biological tissues after death, with researchers expecting a gradual decline in gene activity. Instead, they found the opposite in certain genes, a burst of activity that seemed to defy the very nature of death. This unexpected activity suggested a new complexity to the process of dying, indicating that it is not a singular event, but a gradual process that affects cellular functions in varying degrees. The activation of these genes post-mortem could be likened to the last whispers of life, echoing through the cells in a final attempt to maintain integrity or respond to the stress of cessation. Among the activated genes were those involved in inflammation, immune response, and even some associated with cancer, which added layers of intrigue to their study. The activation of these genes raised questions about their purpose, suggesting that they could be part of a hitherto unknown mechanism of cellular preservation or a biological reflex to the ultimate stressor, death itself. The implications of this discovery are profound, extending beyond academic curiosity to practical applications in forensic science, where understanding the timeline of gene activation post-mortem could significantly refine the estimation of time of death. In the medical field, particularly in organ transplantation and the preservation of biological tissues, this knowledge opens up new avenues for enhancing the viability of organs post-extraction. Moreover, the discovery of zombie genes invites us to reconsider our definitions of life and death at the molecular level. It challenges the notion that death is a clear-cut moment, instead presenting it as a transitionary phase where cellular and genetic activities continue to unfold, albeit in a different context. This shift in perspective not only enriches the scientific understanding of death, but also touches on philosophical and ethical dimensions, prompting a re-evaluation of how we conceptualize the end of life. In essence, the discovery of zombie genes has peeled back a layer of the mystery shrouding life's final moments, revealing that even in death, there is a semblance of life. As research progresses, the continued exploration of these genes holds the promise of unlocking further secrets of life, death, and the intricate dance between the two, offering insights that could revolutionize our approach to medicine, forensic science, and our understanding of biological resilience. The term zombie genes captivates the imagination, evoking images of the undead rising in the realm of the microscopic. Yet, beyond the vivid metaphor lies a profound scientific inquiry into the nature of life and the persistence of cellular activity beyond death. Understanding zombie genes requires delving into the molecular afterlife, where certain genes defy the cessation of biological functions, becoming more active in the hours to days following death. This intriguing phenomenon suggests that the demarcation between life and death at the cellular level is far more nuanced than previously thought. Zombie genes do not represent a singular entity, but encompass a diverse array of genes that are implicated in various biological processes such as inflammation, immune response, and stress. Their activation post-mortem is not random, but appears to be a highly orchestrated event, potentially serving multiple purposes. One hypothesis is that the activation of these genes is a form of stress response. Even in death, cells attempt to repair damage or counteract the stressors that led to the organism's demise. This could be seen as a final, albeit futile, effort to preserve cellular integrity. Another perspective considers the activation of zombie genes as a mechanism to curb the spread of infection post-mortem, a biological legacy that protects the surrounding environment or species by limiting the viability of pathogens after the host's death. Furthermore, the behavior of zombie genes challenges our understanding of what it means to be alive at a cellular level. Traditional views of life and death consider the cessation of heartbeat, breathing, and brain function as definitive markers of death. However, the post-mortem activity of zombie genes suggests that cells retain a level of functionality and can continue to express genes in a meaningful way, blurring the lines between life and death. This phenomenon also sheds light on the resilience of biological tissues and the potential for cellular and genetic activity to persist beyond the expected boundaries of life. It raises fascinating questions about the longevity of biological processes and how they might be harnessed for medical and scientific advancements. For instance, understanding the mechanisms that allow these genes to activate post-mortem could lead to innovative approaches in preserving organs for transplantation, extending the viability of tissues for medical research, and even novel treatments for diseases characterized by similar patterns of gene expression. Moreover, the study of zombie genes opens a window into the evolution of cellular mechanisms designed to respond to extreme stress. It prompts a re-evaluation of our understanding of cellular death and survival strategies, offering insights into how cells navigate the boundary between life and death. This exploration not only enriches our fundamental knowledge of biology, but also has the potential to inform the development of therapies that mimic or modulate these post-mortem gene activations for therapeutic purposes. In essence, understanding zombie genes is not merely an academic exercise, it is a journey into the heart of biological mystery. It challenges preconceived notions about the finality of death, offering a glimpse into the cellular processes that linger in the shadow of life, and highlights the incredible complexity and adaptability of life at the molecular level. As we unravel the secrets of these post-mortem genes, we stand on the brink of unlocking new frontiers in medicine, forensic science, and our comprehension of the life-death continuum. The discovery of zombie genes and the subsequent understanding of their post-mortem activity have profound implications that ripple through the fields of science and medicine, challenging existing paradigms and opening up new avenues for research and application. This intriguing phenomenon not only enriches our understanding of cellular processes at the brink of life and death, but also holds the potential to revolutionize practices in forensic science, organ transplantation, and even the treatment of diseases. In forensic science, the activation of zombie genes offers a novel lens through which the time of death can be estimated with greater precision. Traditionally, the determination of time since death has relied on physical indicators and environmental factors, which can often be imprecise. The post-mortem activity of zombie genes, however, provides a biochemical clock that continues to tick after death, offering clues to the timeline of death that were previously inaccessible. By understanding the patterns and timelines of gene activation, forensic experts can refine their estimates of the post-mortem interval, enhancing the accuracy of criminal investigations and judicial processes. The implications for transplant medicine are equally transformative. Organ transplantation is a race against time, with the viability of organs being a critical factor in the success of transplants. The resilience of certain tissues, as evidenced by the activity of zombie genes, opens up possibilities for extending the life of organs outside the body. By harnessing the mechanisms that allow these genes to function post-mortem, medical researchers could develop new preservation techniques that keep organs viable for longer periods, expanding the window for transplantation and potentially saving more lives. Moreover, The study of zombie genes illuminates the complex processes of cellular stress response and repair mechanisms, offering insights that could be pivotal in the treatment of various diseases. For instance, understanding how certain genes are activated in response to the extreme stress of death could inform the development of therapies for diseases that involve similar stress responses, such as cancer, neurodegenerative disorders, and inflammatory conditions. By mimicking or modulating these gene activations, new therapeutic strategies could emerge, targeting the underlying mechanisms of these conditions with unprecedented precision. Beyond these direct applications, the implications of zombie genes extend to our fundamental understanding of life and death at the molecular level. This research challenges the binary view of life and death, suggesting a continuum of cellular activity that does not adhere to the abrupt boundaries we traditionally recognize. It prompts a reevaluation of what it means to be alive from a biological perspective, encouraging a deeper exploration into the essence of life, the process of dying, and the potential for lifelike processes to occur beyond the point of death. In essence, the study of zombie genes represents a frontier of scientific exploration that bridges life and death, with the potential to impact various aspects of medicine and science profoundly. As we continue to unravel the mysteries of these genes, we are not only expanding our understanding of the biological processes that operate at the edge of life, but also opening up new possibilities for enhancing human health and unraveling the mysteries of life itself. The journey into the world of zombie genes is just beginning, promising a future where the lines between life and death may blur, but the opportunities for scientific and medical advancement become clearer. The exploration of zombie genes, while offering a tantalizing glimpse into the post-mortem landscape of genetic activity, also presents a labyrinth of future directions and challenges that researchers must navigate. As we stand on the precipice of this burgeoning field, the journey forward is both exhilarating and daunting, with the promise of groundbreaking discoveries and the inevitability of complex ethical, technical, and conceptual obstacles. One of the primary future directions is the comprehensive mapping of zombie gene activity across different tissues and conditions. This endeavor requires a multidisciplinary approach, combining genomics, proteomics, and bioinformatics to chart the temporal and spatial dynamics of these genes after death. Such a detailed atlas would not only deepen our understanding of post-mortem cellular processes, but also illuminate the diversity of cellular responses to death across different organ systems. The insights gained could revolutionize organ preservation techniques, forensic methodologies, and our understanding of cellular resilience, potentially leading to novel therapeutic strategies for diseases characterized by similar stress responses or pathological gene activations. However, this journey is fraught with technical challenges. The precise measurement of gene activity post-mortem necessitates advancements in sensitivity and specificity of detection methods. Post-mortem samples are often subject to rapid degradation and contamination, which can confound results and lead to misinterpretation of data. Developing robust protocols that can accurately quantify gene expression levels in these challenging conditions is crucial for the field to advance. Beyond the technical, the study of zombie genes encounters conceptual challenges. The phenomenon blurs the lines between life and death, forcing scientists to reconsider traditional definitions and understandings of these states. This reevaluation may have profound implications for medical ethics, particularly in contexts such as organ donation and the determination of death. As research progresses, it will be essential to engage in interdisciplinary dialogues with ethicists, philosophers, and legal experts to navigate these complex issues and ensure that the exploration of zombie genes is conducted with sensitivity and respect for the deceased. Furthermore, the ethical considerations surrounding research on post-mortem genetic activity are significant. Consent processes for the use of tissues after death, particularly for research that could challenge or extend our understanding of life and death, require careful consideration. Balancing scientific curiosity and respect for the deceased and their families is paramount, necessitating clear communication, ethical oversight, and sensitivity to diverse cultural and personal beliefs about death and post-mortem research. In addition to these challenges, the potential medical applications of zombie gene research, such as novel therapies and organ preservation techniques, must be approached with caution. The translation of basic science findings into clinical applications involves rigorous testing for safety and efficacy, navigating regulatory landscapes, and considering the societal implications of such advancements. In conclusion, the future exploration of zombie genes offers a rich tapestry of opportunities to advance science and medicine, intertwined with a complex array of challenges. As researchers chart this unexplored territory, the collaboration across disciplines, along with the thoughtful consideration of the ethical, technical, and conceptual hurdles, will be essential. The study of zombie genes not only promises to expand our understanding of life and death, but also holds the potential to transform medical practices and challenge our deepest philosophical beliefs about the finality of death.